Hi, in this video abstract I will show you how with my colleagues Rostislav and Diego Bustos we employed an in silico approach to decipher the opening mechanisms of 14-3-3 proteins. But what are 14-3-3s? They are a family of proteins formed by 7 paralogs that present different tissue and subcellular distributions. 14-3-3s are involved in many cellular processes through direct interaction with different partners, including enzymes, transcription factors, cytoskeleton, and apoptotic proteins, among many others. Their misfunction could lead to diseases like cancer or neurodegeneration, making 14-3-3 a very appealing druggable target. Employing molecular dynamic simulations, we studied the plastic behavior of 3 paralogs. Microsecond trajectories allowed us to observe the opening and closing events of the amphipathic groove, a highly conserved binding site of the different 14-3-3 physiological partners. Compared to epsilon and gamma, the zeta paralog was characterized by a higher opening rate. So, we tried to explain the atomic mechanisms underlying these differences. For this, we selected different correlation-based tools to analyze our data. Principal component analysis helped us to characterize the main motions leading to the opening and closing of the binding groove, letting us see a bear trap-like behavior, with rigid central helices acting as hinges for the mobile terminal helices. We also recognized key residues involved in these conformational variations by measuring their displacement correlation through trajectories. To do this, we employed the shortest path map method and observed how residues that moved in a concerted fashion belong to the binding site but also to the opposite side of the protein. Focusing on these residues, we observed how they form a small cavity that presents a lower residue conservation between paralogs that has not been reported to date. Visually inspecting this new cavity on zeta paralog, we observed how, when the binding site opened, a transient aliphatic pi interaction occurred between a phenylalanine residue located at the gates of the cavity and an alanine residue at its bottom. We evaluated this distal opening regulation hypothesis by designing different computational mutants. One of them consisted of a double mutant where the previously mentioned phenylalanine and alanine residues were mutated by cysteine. By this, we fixed the interaction between these 2 residues, forming a covalent disulfide bond and testing how this could stabilize open conformations. As expected, the 14-3-3 mutant opened its binding site irreversibly. Our in silico studies identified that 14-3-3 paralogs differ in their opening rates. We also propose a distal opening mechanism stabilized by an aliphatic pi interaction. These residues are located in a cavity described here for the first time. We are confident that these results will pave the way for developing novel therapeutics for 14-3-3s via allosteric and paralog-specific mechanisms.